Hi everyone, I'm Lucy Zhang from Honor Independent Research, and my research topic is assessing the possibility of using entomopathogenic fungi for mosquito control in Hawaii. My inspiration for this research topic actually came from a pretty weird place. It's a YouTube documentary by National Geographic on the zombie parasite fungus that takes over insects through mind control. If you have seen the video, it's like something out of a horror movie. But then I started thinking, if this fungi can control the mind of insects, maybe they can also be used to control mosquitoes in Hawaii. And that's when the idea for my research was born. In my research, I learned that all mosquitoes in Hawaii are in fact invasive. These mosquitoes pose a significant threat to the ecosystem, which has evolved over millions of years without the presence of those insects. In fact, the decline of forest birds in Kauai Island shows a correlation with the presence of avian malaria, which is a disease that is transmitted by Culex mosquitoes. According to Hawaii Department of Health, the government is currently implementing mosquito traps to control adult mosquitoes while using chemical insecticides and BTI, a biocontrol method with bacteria, as means of controlling the mosquito larvae. Traps are placed in areas where these mosquitoes are known to breed. This method has shown to be effective in reducing the mosquito population in targeted areas. And chemical insecticides and BTI are also applied to standing water where mosquitoes lay their eggs. Once the eggs hatch, the insecticides prevent the larvae from developing into adults. It is important to note that these methods have their limitations. For example, the use of chemicals can have adverse impact on other wildlife in the region. On the other hand, the insect traps have high cost of maintenance and a limited range. While BTI is a novel vector control method, the bacteria need to be ingested by larvae in order to cause infection. An alternative method for controlling mosquito population is the entomopathogenic fungi. The fungi diseases can affect many insects and cause large-scale outbreaks that dramatically reduce their populations. Almost all types of insects can get infected with these diseases. When I was first learning about this fungi, I thought it might be easier to find them in seawater. But after chatting with my mentor, Professor Chen Zhiqiang, I was surprised to find out that it's actually pretty common. And he provided me with this map where he discovered the entomopathogenic fungi that is used in his lab. In addition, I learned that this fungi is actually ubiquitous. In fact, the fungi that is used in my experiment is from the Aloe Dog Park right beside our school. Some other studies have also been conducted on this fungi on the other organisms like beetles. For example, Professor Chen conducted a field study to investigate the impact of entomopathogenic fungi on coconut beetle, a pest that caused significant damage to the coconut palms. Entomopathogenic fungi can infect mosquito hosts by penetrating their cuticles. Since the fungi is naturally occurring, this can be an environmental friendly and sustainable alternative to the traditional methods. On the other hand, it is easier to infect the host compared to the ingestion mechanism of the bacteria method. However, no one has studied the fungi on mosquitoes in Hawaii yet. So this led to my research question: can entomopathogenic fungi be used for mosquito control in Hawaii? And this question can be separated into two parts. The first part is, can entomopathogenic fungi be isolated from Hawaii soils? And the second part would be, is the fungi isolated effective on mosquitoes? This is the overall timeline for my project. I started off isolating the fungi from soils. Then I added the mealworms into containers with soil. Once the mealworms are dead, I put them into petri dishes. In parallel, I did PCR for identification of species. After I obtained a peer culture, I collected fungal spores and quantified them. After that, I tested fungi on Culex larvae to get the final data. Insect-based method is used for this isolation of fungi. Soil was collected from various sites in Honolulu to gather a diverse range of fungi. These soil samples were then placed into containers, and mealworms are added to the containers as bait for the fungi. Over time, the mealworms are killed, and a few days after the death of the mealworms, a visible white and green fungal growth could be observed on the surface of their skin. This is a sign that the fungi exist in the samples. In the experiment, PCR with ITX1 and 4 is used to amplify the internal transcribed spacer region for the identification of fungi. This process involves adding a DNA template, such as the extracted fungal DNA in this case, to a reaction meter containing the primers, nucleotides, and enzymes. The primers are designed to specifically target a segment of DNA. The polymerase then replicates the DNA segment in a process of heating and cooling cycles, resulting in millions of copies of DNA for the same segment, which can then be used for DNA sequencing to identify the species or to obtain other genetic information. This chart shows my basic experimental design for the testing of fungi on mosquito larvae. The duration for each trial is 6 days, and a total of 5 trials were conducted. Every experimental group was paired up with a control. The control is just 25 Culex larvae living in a tube with ultra-pure water, and 2 experimental concentrations were tested. The lower concentration is 10,000 corneta per milliliter, while the higher concentration is 100,000 corneta per milliliter. To begin the process, I scraped the fungal spores from the surface of petri dishes and collected them in a tube with water. Next, I used hemocytometer to count the number of spores. This is a specialized tool that is used to count cells or particles in a sample. Once I had the exact number, I pipetted a certain number of spores into solutions to create different concentrations. Then I added 25 mosquito larvae into each solution. In the experiment, I exposed 4 instar Culex quinquefacientis, which means the final developmental stage of the Culex mosquito larvae, to solutions containing the entomopathogenic fungi, Matarisia, and Sawfly. I used different concentrations of this solution to see how it affected the survival of larvae over time. In this graph, the gray line is the control, the blue line is the experimental group with lower concentration, and the orange line is the group with higher concentration. The lines on the graph shows the average survival rates with error bars of 1 standard error above and below the mean over a period of 6 days. As we can see, an increase in concentration expedited the infecting process greatly. I used the data collected to calculate LC50 of the fungi. LC50 is a measure of concentration of a substance that is required to kill 50% of the test population. In this case, LC50 is a concentration at which half of the mosquitoes will die after 3 days of exposure. And for this experiment, the LC50 value is 3.16 times 10 to the 4th power corneta per milliliter, meaning that this concentration will be able to kill half of the larvae in 3 days. Another study conducted with the same species of fungi on beetle larvae shows a similar trend of increasing mortality with an increase in the concentration of the fungal solution. In addition, the LC50 for this study is also somewhere between 1,000 corneta per milliliter and 10 to the 5th corneta per milliliter as shown on the graph. The fungi discovered in this experiment, which is Matarisia and Sawfly, is one of the optimal insect control agent because it is easy to produce and non-pathogenic to plants and mammals. In conclusion, this study successfully isolates the entomopathogenic fungi from 20 soil samples, and the presence and amount of the fungi varied by location. The study showed that Culex larvae are susceptible to Matarisia and Sawfly. Moreover, the larval stage of the mosquito is one of the most most crucial and effective stage for biocontrol agents, and their lifespan is shown in this experiment to be greatly reduced if contacted with the spores. Although the efficiency of chemical pesticides still outweighs that of the fungal biocontrol agent, the fungi provides more benefit in the long run and making the mosquito control process more sustainable. The susceptibility of mosquitoes to entomopathogenic fungi can also vary depending on the combination of fungal species and mosquito species involved. In this study, I only tested the host pathogen interaction between one fungi and the Culex mosquitoes. To address this limitation, future research could focus on identifying the most effective combination of fungi and mosquito species for the maximum impact. This may involve screening multiple strains of fungi against different species of mosquitoes. This research has important implications on public health and ecosystem conservation efforts in Hawaii. For example, mosquitoes are known to transmit diseases among Hawaii's native bird species. By controlling mosquito, we may be able to reduce the incidence of avian malaria and to protect the native bird population. For the future research students, one of the most important pieces of advice is to stay on top of your schedule. Research can be a very demanding and time-consuming endeavor with numerous tasks and deadlines to keep track of. So to stay organized and avoid falling behind, it's essential to create a detailed schedule and to stick to it as closely as possible. Another important piece of advice is to take initiative and be proactive. This may involve seeking out opportunities for collaboration, developing new ideas and hypotheses, and seeking out new sources of information and inspiration. I want to thank my mentor, Dr. Chen from UH, for your help and guidance with my entomopathogenic fungi project this year. And also Dr. Chen, Ms. Kobayashi, and Mr. Hill for helping me to capture the mosquitoes and buying me mealworms. And Mr. Browning for allowing me to put up mosquito traps in front of his balcony. And last but not least, my parents will always follow up on my progress on WeChat. Thank you for listening.